Thank you very much. We've got about 10 minutes for questions, if anyone would like to come to the microphone. Brian.
Brian Davidson, Royal Free. Can I just ask you a few technical things about the precision slice model? I think it's a great model. We've tried using it in pancreas, but not in biliary tract cancers yet. The first thing I have for you is the slicing. So you take a core, you've sliced it, and you've got 20 sections in slide plates. How do you determine, first of all, what tissue you've got at the outset before you put it into the plates? And what sort of variabilities do you have between the first and last slice? And how do you know that what you're testing in the first slice could be cancer and the last slice could be a bit of fibrous tissue? So how do you deal with that simple, practical problem, first of all?
Well, I think you can't really deal with it, is the answer, without just doing multiple repeats, which is what we do. So we'll do multiple repeats, technical replicates, and do immunohistochemistry staining as well on the samples that we collect to see and try and...
Sorry, is that multiple cores that are the repeats? So 20 slices from each core, and then you do 3 whole plates with...
Yes, slices. Yeah, and we try and let the slices... We put... well, it's either random or haphazard, depending on how you want to sort of define it. We let the slices sort of settle in the buffer reservoir. We'll do multiple cores, and then we can then plate out at random. You could track every slice going through a core, depending on how you've cored it. We don't do it that way, but you can do it that way.
Yeah, so the only way to know whether each slice is going to have tumor cells in it is we find out after using immunohistochemistry and H&E staining as well. So, yeah.
Okay, my second question is about preserving the tissue, because you mentioned that you had viability up to 15 days. Have you got a special technique for the preservation and the composition of your culture medium, or do you just use standard culture medium, and what evidence is there for what you should use?
It's we follow the Kenneson protocol. It's a STAR protocol, and we just do what they did. And it's, yeah, the normal liver and both hilar and intrahepatics. We've done CRLMs, we've done an SCC. HCCs. Well. They're all fine for 15 days. Yeah, just follow the Kenneson protocol.
Yeah, it's the exact same thing, and then we use the EGF as well, human epidermal growth factor, supplemented into the complete media, which is what they use as well.
Yeah. Okay. And my last question for you is about you're presumably using flow cytometry to characterize your immune cells in the samples. Do you then produce tissue lysis to do the flow cytometry, and therefore do you then have to abandon those samples, or have you got any techniques for getting sequential immune responses over time, which is obviously what you want?
I'm using mass cytometry, which is a larger panel than flow, so it's a similar technique, but a much larger panel. It's sort of 35 markers rather than like 7 or 8 that you can use in flow. So the sample is destroyed after you've used it, because you have to dissociate into a single cell suspension. We are looking at the idea of using Hyperion analysis, which is sort of for the spatial version of the mass cytometry that I'm doing. But again, you'd use up the sample. So the way that we're going to try and tackle, try and track changes over time, it would be doing multiple repeats at multiple time points, and then accepting that, yes, slices are going to be different, but we're trying to get an overall change.
And I think also the benefit of using mass cytometry instead of flow cytometry is the fact that you can actually store the samples after you've stained them, whereas with flow you have to do it straight away, basically.
So yeah, we'll get the samples quite late sometimes, so we can prepare them up to a point and freeze them, and then stain and run them all at the same time. So we've actually managed to reduce the amount of batch variation we get between our patients as well, which is quite good, and a sort of a reason for using the mass cytometry.
Good. Okay. Thank you very much.
Thank you.
Sheila.
Can I ask a couple of technical questions as well? So, you've used 250 microns, but can you go a lot thicker? Are they surviving a lot thicker?
Yeah, we've done this in healthy bile ducts. So we cut healthy bile ducts a little bit thicker. It was 280 microns. Yeah. And we impregnated it with agarose gel, to keep the duct open and sliced it, so it just kind of looks like an onion ring almost. And we've kept that cultured for, we only did it for a week, but it was fine.
Yeah, we
at
could
a
probably
week.
go longer.
And we've done thinner as well. So the work that Laura was talking about with the bioenergetics, they're sliced at 210. So protocols have documented in lung tissue different thicknesses, so you can play around with it, but we go with 250 because that's what's been published.
Okay, you answered my second question, which was, have you done it with healthy bile ducts?
Yes, yeah, we have.
As
yeah.
well?
Yeah.
So that's great.
Yeah, it's a little bit harder to obtain those samples because it would have to be from a hilar resection, but we're sort of very mindful of the margin that needs to be kept intact, so it's at the sort of surgeon's discretion that we can get those.
Okay, thank you. And I just have one question for about the cilia.
Yeah.
That was a really exciting study, really interesting. So do you think that any of the mitotic apparatus, the kinases, are all of them interacting with this still protein? And can you use any of those inhibitors?
So thanks for your question. We don't know if there are also other CDK that are involved, that are bind still. So there are a lot of protein that bind still, like CDK1, PLK4, and also Sus6. But many of them are involved in the procentral assembly, so in the early phase of the cell cycle, not in the mitotic phase.
Do you think many of those others, like Aurora kinase, all of those?
Maybe yes.
As well, to regulate the
Maybe many of them are crucial to this point, so maybe yes.
Okay, I have a final question for Emmanuel, maybe, which is, did you say that, I might have missed it, did you say that you're looking for mitotic errors, or is that something that you might plan to do, or is it?
Yeah, in our lab we study these two structures, cilia and sensome, that are more correlated, because so the cilia are present on the surface of the cells only in the quiescent phase. So in the tumor, the cilia are not present because maybe the cells are always in the always in the
in your knockout cells. And
yeah, maybe
compared to the wild type.
we want to identify if there are the correlation between these two organelles and which is the pathway that are correlated to these 2 points.
Right. Any other questions? If there's
Can I ask
you
one
go.
question? Yeah. So it's a practical question about the fresh sampling. And actually, it links a little bit to Brian's points about quality assuring input material. I should remember this, and apologies. Who is it that's sampling? Is it the surgeons, not Tim?
It would be great to have the pathologist with us there,
Yeah.
but it's not always feasible to do that.
And is it always possible, so the perihylers, particularly if they have a non-mass forming growth, if they're periductal infiltrating, even when they're fixed in front of me, I don't know what's what. Are they still being sampled? And if they are, how do you know you've got malignancy?
If they're quite difficult to sample, we'll usually get quite a small bit of sugar, which we can also embed and then slice. They're a bit sort of smaller slices and a bit scrappier than sort of the bigger slices we'll have, but you can usually tell from the viability if a slice gives no viability, it's just completely fibrotic. That's usually a way that we can work out what is going on and which ones to take forward.
Yeah, thank you.
Okay, thank you everybody. I think we should change the name of this session from the fast to the fantastic. So thank you very much for the talks, very good. And I think we're allowed to take a short break now for some tea or coffee, and we start again in 15 minutes. Thank you.